Okay, all right. So how's everything, everybody? So I think starting from last week, you already you already sit for your test one, right? Isn't it? Starting last week, or you have any papers? Did you have any papers? Any test one on the last week? No. So perhaps you're gonna have um many um test one on within this week, right? Isn't it? Or perhaps next week. I'm not sure if next week is still allowed to have to sit for test one. Um, okay, so for test one for the subject is going to be carried out uh, on Wednesday. Okay, on Wednesday at 2:30. Okay, at Makmal Pengajaran 1 and 2 lah. Okay, so okay, so today we are going to continue on the topic amino acids anabolisms, yeah, amino acid anabolisms or amino acid biosynthesis. So as for recap, we have discussed on the pillar. We have discussed on the pillar or the basic cycle that involves for the amino acid biosynthesis that involve three main cycles. Okay, in the pathway. So the first thing is glycolysis. Okay, the second one is citric acid cycle, and the third one is pentose phosphate pathway. So all this together, somehow some of the amino acids they just go through one of the pathway or some of them, yes, a few of them, they are going to have to go through a few cycles. Okay, they're going to go through a few cycles in order to complete the synthesis of the amino acids. Yeah. Um, and we have discussed as well on the there are 20 common amino acids that we have. Okay, common amino acids that we have, and we're not going to discuss all of these amino acids. Basically, on the in their biosynthesis, okay, because there are 20 amino acids that available for each one of them. Okay, each of these amino acids they have their own pathway. Okay, to the synthesis and to how to synthesize them. Okay, so different amino acids they have a different pathway. But then it's very important for you to memorize the each of this each of this structure of these amino acids. Okay, and what is the properties of these amino acids, each of them. And we also we have discussed as well on the non-essential and essential amino acids for humans. Okay, and what does it means on the what is the non-essential and what are the amino acids? What is meant by non-essential and essential amino acids? So we have discussed on that one as well. Okay. Okay, so these are the citric acid cycle, glycolysis, and pentose phosphate pathway that involved together in the amino acid biosynthesis. So these are the common metabolic intermediates that can be found in these cycles. Okay, and they will involve to the synthesis of the amino acids. And for each of them, for each of the amino acids, they are going to require nitrogens, which supplied by the glutamate and glutamine synthesis. Okay. And yeah, so during the synthesis of the amino acids, there will be a process to assimilate nitrogen to the biomolecules, where in this process, it will involve the incorporations of the ammonia, okay, in a form of NH4 to amino acids. So we know that where does this nitrogen comes from, because we have learned on the nitrogen cycle during the first topic that we have discussed before, right? We did discuss on the nitrogen cycle and how the this nitrogen um, will be absorbed by the organisms in a form of ammonia, okay, which is a form of this soluble nitrogen that can be absorbed to the organisms. So this is how the ammonia, the source of it, is going to be used to this formation of the amino acids. Okay, and in the same time, um, glutamate and glutamine is also play an important role to help for the assimilations of the source of the nitrogens to these biomolecules or other amino acids. Okay, and in this case, alpha amino groups, okay, alpha amino group, which is NH3 or NH2 in a form of alpha amino group in the any amino acids, comes from these alpha amino groups of the glutamate via the transamination. Okay, and glutamine, however, it will provide nitrogen side chain in the synthesis of few important compounds. Okay, so there are examples of non-essential amino acids and the precursors that involve to the formation of these amino acids. So we know that during the amino acid catabolisms, okay, amino acid catabolisms which involve the alanine, aspartate, and serine, before this, they are actually the degradations of these amino acids actually comes from and intermediates. Okay, so the same thing goes to these anabolisms. So, so the formations of this alanine, aspartate, and then serine, for example, here, they actually derived from the same intermediates. Okay, derived from the same intermediates. So remember when we talk about the transaminations, so the transamination is actually a reversible. Okay, a reversible. It can go back to um, uh, it can go to either way, okay, both ways, but it depends on the um, um, metabolic requirements. Okay, so that's why in this case, when we want to synthesize back the amino acids, so we have to go through with the same intermediates or precursors. So in this case, for alanine, we have the pyruvate. Okay, we have the pyruvate, and for aspartate, we refer to the oxaloacetate, and the serine in this case, we have the 3-phosphoglycerate. Okay, and take for example, for those amino acids which is essential amino acids, which we can get it from our diet. For example, methionine, histidine, and tryptophan. So each of them, they come from the oxaloacetate, or in the case of the methionine, and ribose 5-phosphate in case of the histidine, and phosphonyl pyruvate plus the erythritol 4-phosphate in case of the tryptophan. Yeah? So in case of the tryptophans, you can see they involve two important precursors, because if you can see how the tryptophan is formed, they have a little bit complex structure compared to the other amino acids, which they have this double ring here. Okay, the first and second ring, and they have the other extensions to this carbon skeleton. So this is tryptophan. So it's a little bit complex. So it makes sense if it's actually derived from two precursors. All right. So biosynthesis of the essential and non-essential amino acids. So to refer which which precursors and which amino acids that actually derived from which precursors or intermediates. So we can actually categorize each of these amino acids based on what are the precursors or intermediates that they actually derived on. Okay, so for example, we have alanine, valine, and leucine. So they are derived from the pyruvate, okay, which we can actually synthesize them from the pyruvate. And in case of the asparagine, methionine, threonine, lysine, and leucines, we can get it from the oxaloacetate, whereby the oxaloacetate actually will be we go through the process of transaminations and we get aspartate, and then this asparagine, methionine, and then the other amino acids actually derived from the aspartate itself. And these those amino acids with the star is actually those amino acids which is essential. And for alpha ketoglutarate, they are a very common precursors. Okay, very common precursors. Even we already is quite familiar for us because we have discussed on the transamination, and the alpha ketoglutarate is the a very important intermediate as well during the transamination process. And in this case, I think it's easy for you to relate to the formation of the glutamate, right? Because during the formation or during the transaminations, we have the product of glutamate, and from there we have the glutamine, proline, and arginine as the other. We can produce the other by these amino acids. And we have phosphonopyruvate and erythritol phosphate. They can process to the phenylalanine, and then phenylalanine it can degrade to tyrosine. And I think this is also quite familiar, as we have discussed before about the disease, PKU, which is there is the problem with the absence of the enzymes which make it the patients with the PKU cannot synthesize phenylalanine to the tyrosine. So as a result, there is the accumulations of the phenylalanine in the blood, and that is why the reasons they get the PKU. Yeah, so this is the so this is where the tyrosines and phenylalanines they actually can be synthesized from this precursor phosphonopyruvate plus the erythritol phosphate. And in case of the tyrosine, they actually can be processed directly from these precursors, and also tryptophan as well. And for ribose 5-phosphate, we have these only one amino acid actually derived from this ribose 5-phosphate, which is histidine. And we have the 3-phosphoglycerate, which can be which can be synthesized, which can be which this precursor can synthesize the serine, and then the serine can synthesize to the cysteine and the glycine. All right, so let's take a look at the first one, which is the formation of the glutamine, proline, and arginine. So in this case, for the formation of these four amino acids, so they are synthesized by using the asparglutarate as the precursors, and as I mentioned before, the assimilations or the involvement of the ammonia or the source of the ammonia or nitrogen into glutamate will require two reactions. Okay, require two reactions. So which is the first one is the glutamate will react with the ATP. Okay, so in this case they need the presence of the ATP, and they get the gamma glutamate phosphate plus ATP. And the second one is the alpha glutamate phosphate will assimilate with the first one. Okay, assimilate with the first one, which is the ammonia. Okay, with the ammonia and with the presence of the glutamine synthase, then we get the glutamine as the amino acids as a product. And so as the summary for these equations, okay, starting from we get the glutamate and we have the ammonia, okay, and plus the ATP, we get the glutamine as the product of these amino acids from the glutamate. And proline also is the derivative of the glutamate, where it can go through some arrangement, okay, in case of the glutamate. So and we can get proline out of glutamate. Okay, so for all organisms, they can assimilate, including human, animals, all organisms. We can assimilate the ammonia through the reactions that produce the glutamate, carbonyl phosphate, and asparagine. And so, in case of the biosynthesis of the glutamate in bacteria and plants, glutamate is produced from glutamate, okay, in a reactions catalyzed by glutamate synthase. So this is in bacteria and plants. And alpha-ketoglutarate is an intermediate, okay, that can be found in citric acid cycle, where in the process the alpha-ketoglutarate will go through or undergo the reduction nations, where the glutamine will act as a nitrogen donor. So in this case, it's actually the same thing that we have discussed with it during the transamination process. This is actually transamination process where the alpha-ketoglutarate, okay, will go through this what we call as reductive amination, and then glutamine will actually will give away one of the amino groups to this glutamine. So as the result, then we get these equations here, where out of this process we can get two glutamate. So this is in bacteria and plants. So the formations of the glutamate in bacteria and plants. Hold on a second. I need to charge my laptop. All right. Okay, let's take a look at this biosynthesis of glutamate. Okay, so we have the presence of this glutamine aminotransferase, or this is, of course, this is happened during the amino transamination. So glutamine aminotransferase is the one of the enzymes that actually catalyze the process of the transamination during the biosynthesis of the glutamate, okay, in plants and bacteria. So basically, we have this glutamine aminotransferase, and what happened is that in the catalytic site of the glutamine aminotransferase, so we have the glutamine. Okay, we have the glutamine. So what happened is that this amino group, okay, this amino group of this glutamine will be cleaved from this glutamine, okay, And then so we have the whatever amino acids here, which is the will comes in. It will comes in will become the amino group acceptor from the glutamine. Okay, and then we can get the glutamate. Okay, from the process of the glutamate of this cleavage of this amino group. Okay, we know that glutamate, it can, during the transamination before, every single amino acids, they can actually go through the transamination and we get the glutamate as the core product, right? Core products of whatever transaminations of the amino acids. And we get the glutamate as the product. And in this case, during the process of to get back or biosynthesis of the glutamate, so meaning that here we as the acceptor, it can be any of the amino acids, yeah, any of the amino acids to receive this amino group from the glutamine, and they will be processed uh, to, uh, to 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 get back to this glutamate here, okay, to get back the glutamate, right? So, and whatever the amino acids then will be released, okay. So this is the biosynthesis of the glutamate, and this is the processed biosynthesis of the proline and arginine. So it started with the glutamate, okay, towards this pathway. Um, uh, there will be rearrangements of the glutamate okay, glutamate, and then with the presence of the glutamate dehydrogenase, okay, and then NADPH as the precursors, okay, as the precursors, or in this case, NADPH, as we have discussed before, is play a role as the cofactor, yeah, cofactor, and we get the glutamate gamma aldehydes, okay, where the phosphates from these gamma glutamate phosphates here, okay, will be released, and we get the glutamate gamma semialdehyde since the phosphate is being gave away in the process towards the where uh, uh, in the presence of glutamate dehydrogenase, okay, and rearrangements of the glutamate gamma semialdehyde, and we can get this ring of this part proline, yeah, proline, and we finally get the proline here after they do this kind of rearrangements to the carbon chain of glutamate semialdehyde. And towards this way, it's quite a long pathway to this formation of the arginine. And so, yeah, I just will just highlight a certain pathway or certain process in the to this formation of the arginine. Okay, so we can see towards this way we have one aminotransferase, which is, you know, that this is a process of transamination, right? So you can see here this is the involvement of the glutamate, and we get the alpha glutarate as a side product, and we get an acetylornithine, and this product here, and acetylornithine, will actually uh, combined or contribute to the product that or intermediate that will be channeled into the urea cycle. Okay, as you can see from an ornithine, then we get the ornithine. So because we know that ornithine is one of the intermediate in the urea cycle, so we get the arginine at the end of it, right? Because we have the in the urea cycle, we know that we get the arginine, one of the process or one of the intermediates or products that's involved in the urea cycles. So basically, in the formation of the arginine is very important. The formation of arginine is very important in the urea cycle. Okay, so this is a summary of the formation of the arginine. So it I think it starts here, starting from the glutamate. there are many intermediates that involved. Okay, and in the process as well, there is the involvement of the product, and um, and then keep changing the intermediates, the product, and another intermediate comes in, and then the product. So and finally we get arginine. So arginine is quite complex amino acid as well. So that's why it go through many many process one by one to make it a complete complex amino acid of arginine. Okay, so we know that arginine is listed as the one of the essential amino acids. Okay, but it is considered as the essential amino acids during the growth, during the childhood growth or pregnancy. Because during this period, um, human, especially they, of course, human, we require a lot of arginine in this case, so it is considered as the essential amino acid during this period. Um, and but then for the normal adult, arginine is considered as non-essential because arginine is our body can produce arginine. Yeah, we get enough of it from our body. Our body can produce it, but during this period, during the child growth, um, uh, childhood growth, and also pregnancy. Uh, so these two categories, they need more arginine, which should be supplied from the diet. Okay, so that's why arginine is considered as essential amino acids. So for these categories, and yeah. Okay, so the next one is the biosynthesis of the serine, cysteine, and glycine. Uh, So for the serine, cysteine, and glycines, the precursors that involved to this formation of three amino acids is 3-phosphoglycerate. Okay. So if you can see here, we have the 3-phosphoglycerate go through the formation of the 3-phospho hydroxypyruvate. Okay, 3-phosphohydroxypyruvate, where the uh, we get the reduction of the NAD to NADH. Okay, and in this case, we the part, the parts of the hydrogens at the one of the carbon skeleton is removed to actually reduce the NAD towards the NADH, and we get 3-phosphohydroxypyruvate. Yeah, so 3-phosphohydroxypyruvate then will be it go through the process of transamination in this case to get the 3-phosphoserine. So in this case, the glutamate uh, will play a role to actually give away the amino group. Okay, the amino group, and we get the 3-phosphoserine. Yeah, so we can see here the amino group that actually derived from glutamate itself. So we get the 3-phosphoserine, and in the presence of phosphoserine phosphatase, which is the enzyme that catalyzes the removing of the phosphate from the 3-phosphoserine, and we get the serine. Yeah, we get the serine, and towards this way, at the end of it, we can get the glycines from the serine. And I think we have discussed here how the formation, how the formation or the pathway of the catabolisms of the glycines before the glycine before it started with the um, a glycine, and then we get the serine. Yeah? But then to the synthesis of the amino acids, it started with the serine and end up with the glycine. Yeah. So same thing. We have to involve the PLP and also the presence of the folate. Yeah, folate. And it will go through the transferring. Okay, it will involve the transferring of the methyl group in this case. Okay, because we have also discussed on the how the methyl group is transferred during the last topic of the uh, apa ni folic metabolism, right? Yeah. So so the same thing here goes apply as well, the process, and we get the glycines. So I think this process from serine to glycines is very familiar to you. Okay, and so in this case, in this other side here, in this side we have the biosynthesis of the cysteine. Okay, so we started with the serine, where the serine actually react or with homocysteine. Okay, and then we have the presence of the PLP here. And we get the sister threonine. Yeah, we get the sister threonine out of the formations or the reactions with the homocysteine and serine, and we get the cysteine finally as a product. So meaning that serine is just not only can produce glycines, okay, but then specialized in mammals, serine it can also produce the cysteine. Yeah? Uh, so and this process actually can be relate to the methionine cycle. Okay, relate to the methionine cycle. Okay. All right. So this is biosynthesis of cysteine in bacteria and plants. Because this is biosynthesis, okay, for the cysteine, there will be a different pathway or different process. What we found in human, okay, in mammals or other, yeah, not human lah, of course, other organisms, mammals, um, uh, and for the biosynthesis of the cysteine in mammals, so it's actually derived from the serine and we get the cysteine in this pathway. But then in case of the biosynthesis of cysteine in bacteria and plants, so it applied another pathway. So it different way between the cysteine in the human or animals, which is mammals, and the one in the bacteria and plants. So they applied a different pathway. So in this case, a little bit complex in bacteria and plants. So because they have a different enzymes that involve, and so it has to go through the process of the getting the sulfur, getting the sulfur from this sulfide, from the sulfide, which is derived from this adenosine 5-phosphosulfate or APS. So this towards this process, okay, starting from the ATP release, ATP and the sulfate. So this process here is to actually prepare the source of the sulfur to the sulfide and then turn it into the sulfide. So this is the sulfide that can be that can react to, or it can absorb by this process, which is involved the enzyme coacetylserine lyase. So then we get this system over here for in case of the bacteria and plants. So it's quite complicated as well in the bacteria and plants to this formation of the system. All right. So the next one, we move to aromatic amino acids. Aromatic amino acids. So aromatic amino acids. So we have the phenylalanine, tyrosine, tryptophan, and the tyrosine actually can be derived from the phenylalanines towards to another pathway. And what's the important about the aromatic amino acid is that there is one precursor that is very important is the shikimate. Okay, shikimate. So this is the structure of the shikimate. So how we get the carboxyl group over this ring here, and then we have three hydroxide group yeah, in the opposite side of it. Okay, and this is the shikimate. Okay, with one of the double uh, double bond at this side. And shikimate is very important intermediate to the synthesis of aromatic amino acids. So what happened to this shikimate is that um, so shikimate has to prepare itself to this formation of the chorismate. So you can think of the shikimate like prepare itself or activate itself to the chorismate. Okay, once it turns into the chorismate, then it can go to any pathway that they like towards phenylalanine, tyrosine, or tryptophan. And so there are three steps involved for the shikimate to be converted into the chorismate. Okay, so this is a very important stage to this formation of aromatic amino acids, because without the chorismate, so the shikimate cannot be converted into other amino acids, other aromatic amino acids. They have to be changed, or they have to be converted first to this chorismate before it can synthesize these amino acids. So there are three steps involved for the shikimate to be converted to the chorismates. So the first step is the condensations of the shikimate to this phosphoryl pyruvate. Okay, and then so at the end we get the chorismate. Okay, so this is chorismate. And the biosynthesis of this tyrosine and phenylalanine. So we take a look at this biosynthesis of tyrosine and the phenylalanine at first. So how the once we get the chorismate, so it will be converted to the prephenate here, the chorismate to the prephenate, and then from the prephenate it can be split into the to two different pathways over here, towards the formation of the tyrosine and towards the formation of the phenylalanine. And for the prephenate, okay, to this prephenate, towards this way to the formation of the tyrosine. So the process will involve the presence of NAD, where the NAD will be reduced to the NADH. Okay, and then it also produces CO2, which is actually comes from this carboxyl group of the prephenate. And since this carboxyl group of this prephenate is removed together with the reduction of the NAD plus, then we get this 4-hydroxyl-phenylpyruvate. And during the transamination process here, from the 4-hydroxyl-phenylpyruvate, where the glutamate give away its amino group or becomes the amino group donor, then we get the tyrosine as the amino acids. Yeah? And so on the other side over here, we have prephenates where it can be converted into this phenylpyruvate. Yeah. So in this case, there is no such reduction of this NAD involved. Okay, so but then over here there is the removing of the carboxyl group of the prephenate to this carbon dioxide plus hydroxide, and we get the phenylpyruvate. Yeah, And of course, if you can see, these two process over here is very important to this formation of the 4-hydroxyphenyl and pyruvate and phenyl pyruvate. So this two structure is different over here with the presence of the hydroxide group at this ring, okay, at this carbon. And for phenyl pyruvate, so there is no OH. Okay, there is no OH for the phenyl pyruvate. So that makes this tyrosine and phenylalanine it's different because phenylalanine there is no hydroxide group over here, but over here for the tyrosine we have the phenylalanine, we have the hydroxide group. Okay, so but both process involve the transaminations. Yep. So and in this case, I think it's quite familiar for you as well because we have discussed on the transaminations before, where the whatever the amino acids, the catabolism of them will involve or produce glutamate. So so through this biosynthesis of these amino acids, of course we need back the glutamate in the process as the amino group daughter. Okay. And I think it worth as well to mention again about the tyrosine, where it can is actually a conditional and non-essential amino acids in humans. Okay, and okay, so phenylalanine is actually essential. Okay, phenylalanine is actually essential amino acids because our body cannot produce it, and we have to get it from the diet. All right, we have to get it from the diet. But once we get enough of the phenylalanine from the diet, then our body can produce. Okay, we can actually produce. Our body can produce the tyrosine. Okay, but provided we have the ability or we have the presence of the enzyme phenylalanine hydroxylase in this case. All right. And another one is the biosynthesis of the tyrosines. So biosynthesis of the tryptophan. Biosynthesis of the tryptophan. So tryptophan is quite a complex process. It's a complex process. So it started with the glutamine, okay, glucosamate, where it started with the glucosamate, and it has the glutamine as the donor, okay, as the donor of the amino group. And you can see here from the glucosamate, it can be converted into a tranilate, okay, a tranilate, where this part over here, okay, this carbon here is attached to this carboxyl group, and the CH2 and O here. So it's actually this part is removed, then it actually replaced by the amino group that derived from this glutamine during this process of the transaminations, which involve the tranilate synthase. And since this ring is prepared over here in the formation of the tranilate, then the PRPP will comes in. Okay, will comes in to actually give away the whole group of this, which is attached to this amino group in the tranilate, and we get this N5 phosphoryl tranilate. And somehow this ring has to be rearranged. Okay, has to be rearranged, and this intermediate over here Okay, with the some rearrangement involved, we get a smaller ring over here with the side chain attached to this phosphate. And at the end of it, okay, at the end of it, we have the involvement of the serine, okay, involvement of the serine to complete the process where the serine, uh, where the group, this phosphate group that attached to this carbon chain will be removed to produce the glyceraldehyde triphosphate. But then serine comes in and attached to this ring over here. Okay, ring over here, so we get the tryptophan. So we can say that the serine also is very important uh, uh, precursors as well through this formation of the tryptophan. So serine will come in and complete the process. Yeah. Right. Yeah, I'm not going to discuss step by step in very details through this formation of the tryptophan because it's very quite complex. But then, if you're interested, you can find out that okay, each of it how these formations of this uh, phosphate or this ring, how it can be uh, it can be rearranged in this way and so on. It's quite complex stuff. Okay, so for me to explain that one by one, it's gonna be perhaps I need another 2 weeks for it. Okay, all right. So not 2 weeks. Okay, another 2 classes, another 2 sessions. Okay, so okay, here we discussed on the histidine. Okay, histidine. So histidine is another aromatic amino acid as well because it has the one ring attached to it, and what makes this histidine is very unique because it has the it derived from three precursors. Okay, it derived from three precursors, which is the PRPP, purine, and glutamine. Okay, so PRPP will contribute to the 5 carbon, the 5 carbon of the histidine, and purine ring of the ATP will contribute to the nitrogen and a carbon in the histidine, and glutamine will supply the second ring nitrogen. Right? So PRPP is 5-carboxyl 1-pyrophosphate or PRPP. Um, yeah, so this is our PRPP lah. Okay, PRPP, a form of nucleotides, okay, a form of nucleotides. It's very important for this formation of the amino acids. Okay? And so what happened is that the PRPP will react with the amino group uh, at this ring. Okay. So and then ATP is at this side. Okay, and then um, and plus with the second ring nitrogen will supply the glutamine. Okay, but which is supplied by glutamine will attach to this this carbon over here, and we have the uh, chain of this amino group plus the CH that which actually derived from this ATP, okay, attached to carbon chain of the PRPP, okay, over here. So when these three sources combined, okay, rearrange, they finally, we get this histidine. Yeah. So, so uh, based on the figures showed to you, it's actually highlighted. This is actually highlighted where does this each of these components comes from, okay, which is in pink shade and shade pink. So meaning that these components over here derive from PRPP, yeah, derived from PRPP. So actually, you can see PRPP is actually a ring like this, and it has the 2-phosphate over here. But then during the process to these formations of the histidine, then it reacts with the ATP, a part of the ATP and this glutamine, that it makes some rearrangement. So that's why the complex or root of this PRPP ring will be detached. Somehow it will become like this, and it rearrange with the reactions of this ATP. Then it will come out with the uh, a smaller ring which is attached with the elements, some of the elements from the ATP, and include the sources. Okay, include the source of the nitrogens that comes from the glutamines. Yeah, so one and another one here. Okay, and so um, and we also have the another amino acid as well, which is 3 non-essential and 6 essential amino acids. For the pyruvate, alanine, aspartate, and aspartate is actually synthesized from the pyruvate, and oxaloacetate, and the pyruvate, alanine is derived from the pyruvate, and aspartate is derived from the oxaloacetate, as you can see here. And both of alanine and the aspartate, they synthesized from this um, precursors over here, from the process transaminations, from the process of transaminations. And in case of the methionine, threonine, lysine, isoleucine, valine, and leucine, so these are actually the essential amino acids, and their biosynthetic pathway are complex and interconnected each other lah. Okay, interconnected to each other lah, between the valine, leucines, methionine, lysine, threonine, isoleucine. So and I think we are not going to discuss in details for these ones, because perhaps it is not going to go through just in one pathway, like just glycolysis or citric acid cycles, but perhaps it will involve the complex pathway that involves uh, three pathways or other pathway as well. Okay, so let's take a look on the some of the significance of the essential coenzymes. So we know that coenzymes may act as carriers, okay, of specific functional groups, as we have discussed before, like the LP, like many vitamin B derivatives. So they can be considered as essential coenzymes, because without these coenzymes, there is no carrier. Okay, there is no carrier to actually bring uh, a certain element, a certain group of a certain functional group to carry to another intermediate or another carrier. Yeah. So for example, last time that I've mentioned how the PLP actually received, or no, PLP actually received the methyl group from the uh, from the uh, the homocysteine, homocysteines, and then homocysteine is converted somehow to the no homocysteine, homocysteine, okay, and then they become a methionine, right? Then that is actually how the methionine cycle is uh, is actually contribute to this formations of the methionine cycles. And And these essential coenzymes, for example, PLP and another derivative, they actually modified, okay, to fulfill the course of the reactions, and it can be regenerated to use uh, to use in the to go back to the cycle and to keep it in the active form. Yeah, and all the water soluble vitamins, with the exception of vitamin C, are actually coenzymes or precursors of the coenzymes. So any of the all the water soluble vitamins, water soluble vitamins, they actually can actually play a role as the coenzymes or precursors of uh, for the process of the metabolisms. Right. So we have here coenzymes, for example, NADH, NADPH, FADH, PLP, coenzymes A or thiamine pyrophosphate. So all these are very important in catabolisms or anabolism. So, for example, we have the NADH. So NADH is very important in the catabolisms or energy releasing reactions. But then for NADPH is crucial for anabolisms, yeah, and reaction that involve the smaller molecules combining to form a larger complex molecules. So this is the difference between NADH and NADPH. Yeah, NADPH. Um, uh, if we look at the example here, we have the NADH, which are very important for the cellular respiration. Yeah. Yeah, of course, towards cellular respirations, we know that cellular respiration is a form of energy releasing because it's involved the catabolisms of the uh, major molecules, right, to simpler molecules, and in the process, it produces the ATP, and of course, the process involve the energy. In the process, the energy will be released in the form of the ATP. But in case of an ADPH, is very crucial for anabolisms process, and for example, the the photosynthesis. Okay, all right. So this is the example of essential coenzymes. For example, nicotinamide. So this is the oxidized form of nicotinamide. Okay. And this is the reduced form of it. Okay, reduced form of nicotinamide. And so the reduced form of the nicotinamide is the NADH or NADPH. Yeah? NADH or NADPH. And it's participate in the redox reactions, okay, the uh, from this process of transferring the hydride. You can see here we have the H here over here. So this is the hydride uh, that will be released, okay, in case of towards this reduction, uh, and um, and then we will have the it's reversible process lah. Okay, it's reversible process for in case of this oxidized and reduced form. It can go through any way. Okay, it can go in both ways anytime. Depends on the substrate that presents. Okay, and we have another form of essential coenzymes, which is pyridoxine and pyridoxal phosphate or PLP. And I think we have discussed on this previously on the previous topics, and how the ability of the PLP or pyridoxal phosphate can form the stable shift base. Okay, to actually act as a carrier to this amino group from the amino acids, any of the amino acids, and not just the transaminations, but the PLP is very apa kita kata uh, um, very unique because it can not just work in the transaminations, but it can actually act as a carrier for many process like or alpha beta decarboxylation, beta and gamma elimination, respiration, alpha reactions. There are so many reactions that actually use the PLP to do the process. Yeah, and okay, so these are the list of the enzymes that require NAD and NADP in the process of in its synthesis, meaning that this NAD and NADP will act as the coenzymes to actually help the process, help the synthesis for reactions. So, for example, we have the alcohol dehydrogenase. So, alcohol dehydrogenase is a very important enzyme to metabolize the formation of the alcohol, and this is the enzyme that this is the coenzyme that uh, involved, which is NAD plus, and of course in the oxidized form, and if it goes to reduced form, this is the reduced form of NAD plus. So here we can recognize it in the with the presence of the H here, so meaning that this is in the reduced form. And we have more other than this. We have the lactate dehydrogenase, pyruvate dehydrogenase, and ADH dehydrogenase. All these enzymes that with the name dehydrogenase, basically they need the NAD plus. They need NAD plus or NADH, where it can go, because this process is reversible. It will apply NAD plus or NADH, either one. Okay, and the same thing goes to some of the other enzymes over here. Okay, they also need the coenzyme in the form of NAD plus, okay, or NADH. It depends on the nature of the reactions again, because it can go both ways. Yeah, it's a reversible process. Okay, and let's take a look on the some of the functions of the amino acids. So these amino acids, it can act as the building blocks of the proteins. We know that amino acids is just a single a single element that form in the long peptides of the proteins, yeah, or enzymes. Um, so we can say that amino acid is the building block of the protein. So it makes up this a long chain of proteins or the enzymes or functional proteins, and it also acts as precursors for so many functional biomolecules, coenzymes, nucleotides, alcohol, alkaloids, hormones, neurotransmitters. Okay, so and it also can play a role as the cell wall polymers, porphyrins, antibiotics, pigments, and neurotransmitters. Okay, so I've mentioned once neurotransmitters. So these actually needs for these amino acids. Yeah? All right. So one of the important uh, contributions of the amino acid is that the one of it is the glycines. So glycine is a major precursor for the biosynthesis of porphyrins. So porphyrin is very important to actually uh, synthesize the heme product. Okay, synthesize the heme product, and this is the heme how we have the so heme. It's very important to actually hold the metal, okay, in the middle like this, so we can actually find the heme in the synthesis of blood, and banyak lagi lah. Okay, so so the role is to actually to hold the metal in the uh, in this position in the heme, and it's very important for the uh, uh, for the process of reduction and oxidation. Okay, for to in the pathway. So um, so if someone if someone get the genetic defect of biosynthesis of porphyrin, which contain the uh, which actually have this heme, okay, so they will get the porphyria disease, okay, porphyria disease, which is related to this uh, uh, what? uh, red blood cells, yeah, red blood cells here, touch foot lah, okay, um, so there might be of course there will be um, any some defect on the concentration of the heme product. Yeah, so this is the signs, the signs of those that have the genetic defect of synthesis of porphyrin. Um, okay. Yeah. So to the formation of this heme is actually derived from this reactions of the glycine with the succinyl CoA. And actually, for the succinyl CoA, we can find it in the which cycle actually you can find these intermediates of succinyl CoA. Anyone can give me the answer? Which cycle that we actually can find the succinyl CoA?
TCA cycle, Doctor.
All right, TCA cycle, yeah. So succinyl CoA is one of the intermediates in this TCA cycle. So it will react with the glycine, yeah, with the glycines, which is the precursors in the biosynthesis of porphyrin, and they will do this rearrangements towards the structure, and finally get this ring. Yeah? So they will when they restructure it and get into the shape, then they can hold the metals, the iron, the iron with 2 plus, and this is the heme lah. Okay. All right. So the next one is the aspartate, or the amino acids that serve for the structure of bacterial cell walls. And what we know the cell walls of bacteria is the peptidoglycans that contains the alanine and the glutamate. Yeah? So this is how the alanine and the glutamate they arrange in this way. Okay. So the alanine and the glutamate it become a very important structure for the cell wall stability. This alanine and glutamate they actually uh, hold these muramic acids. Okay, they hold these muramic acids, and they have lysines and other amino acids as well to hold it at the bottom part. And we have the glycines over here play a role as the bridge that hold this pillar between these two. Okay, so each pillar they have these the alanine and glutamate that hold these muramic acids over here. Okay, so this when this many parts of this tetrapeptide and interbridge, we have this kind of structure of peptidoglycan of the cell wall in bacteria that we can actually observe under microscope. We can actually see it as this crisscross network pattern like this. So this is what make up the crisscross network pattern because of this formation of this um, uh, interbridge between this pillar that's made up by this alanine and glutamate and amino acids. All right, so this peptidoglycan in the cell wall of the bacteria. And when we talk about the functions or important role of these amino acids, and I think we have discussed on this one, on the nitrogen disposal and excretions during the urea disposal, right? Urea disposal, that will relate to the but that will relate on the oxidative phosphorylation and then go to the process of the removing the ammonia in the urea cycle. And in this case, to is very important. So single amino acids, every single amino acids is very important because we need the to remove the ammonia, the amino group from the carbon skeletons to actually we need to remove it. Our body need to remove it properly to channel it to this urea cycle. And we have discussed as well how the function of the alanine as the amino acids that are very important to actually act as a carrier to bring this amino group, okay, from the muscle to the liver, because alanine it can be absorbed in the blood, yeah. So it's very so the task of the alanine is very important in this case to actually bring this amino group from the muscle and to the liver, and we can remove the amino group uh, um that which is carried by the alanine to 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 the urea cycle, okay, to be removed in a form of the urea. Okay. Okay, so we have discussed this one, and glucose alanine cycle. We have discussed on this one as well. Um, Sorry about it. Okay, so we have discussed this one, right? Muscle. Okay, do you have any questions about this glucose alanine cycle? I think we have discussed about this many times before this, so hopefully you can relate to this apa ni, the transaminations, right? Transamination process, and it involves quite a few elements juga lah. Quite a few elements that we have discussed before about the transaminations, the role of amino acids and the urea cycle, so all inside this glucose alanine cycle. Okay, so so we can say that the amino acids alanine, okay, play a role in many many functions, including, of course, protein synthesis is the most important one, to build up the protein block. Okay, it play important role in the cell signaling, acid-base balance, osmolar regulation, metamorphosis, pigmentations. So many things actually derive from these amino acids. So many pathways. So for the summary, we have the glucose. Okay. So for the summary, there are actually many cycles that involve to the synthesis of these amino acids. Yeah. So these cycles that we have discussed for like pentose phosphate pathway, glycolysis, citric acid cycles. So these three amino acids, these three cycles, or these three pathway, they have their own way how to produce this each of the amino acids. Yeah. For example, this histidine is actually derived from this ribose 5 phosphate or 5P, which is actually um, derived, which is actually this intermediate derived from this pentose phosphate pathway in this stage over here. And then the histidine or the serine is also can be derived from the histidine itself, or and serine can be synthesized from this glycolysis pathway with the 3-phosphoglycerate as the intermediate. And we also have the phosphorylpyruvate or PP, and then we also have the pyruvate. So all these three important intermediates in the glycolysis, they can be a very important intermediate to produce or synthesize a different amino acids. Yeah. So towards this way, then we look into this how the uh, intermediates that involve in the formations of the citric acid cycle, and how these precursors or intermediates derived uh, um, or synthesized. It can be the precursors to synthesize more and more amino acids towards this way. And alpha-ketoglutarate is very important to this formation of the glutamate, proline, and arginine. And we also discussed on the important part of the apa ni, uh, important role of the glutamate and glutamine, yeah, because this glutamate and glutamine is very important to the uh, to prepare the amino acid with its amino group. So we get back the amino group from the glutamine or glutamate to complete the process of the amino acid synthesis, yeah, for each of the amino acids. Okay, I think that's all for this topic. Yeah, that's all for the amino acid biosynthesis. As I said, we haven't discussed about all the synthesis of the amino acids yang complex, all this yang complex and complicated too. We haven't discussed about it, but we have discussed on a few of the amino acids on how it is synthesized. Okay. So any questions so far? Any questions regarding the amino acid biosynthesis on this topic? So we have another, just one more topic on the amino acid biosynthesis, which is it's actually the topic. The topic is a little bit different because another topic that we will discuss on the coming Wednesday and also on the next weeks is regarding on the how the synthesis on the amino acids on the starting from DNA towards RNA, and how this amino acid is become functional functional proteins or functional um, apa ni, another elements lah, okay, after it is biosynthesized. Okay. um, so I'm going to stop share if you don't have any questions. Sekejap, I nak ambil attendance sekejap. So in the meantime, I'm going to wait for your questions if you have any questions. Okay, not just related to this topic, but maybe perhaps you have other questions. In the meanwhile, I will take attendance lah from the participants list ni. Okay. Dua 205 708 ni Sashini kan? Sashini, you there, right? So I think for group B, your lab practical, you have to submit this week, right? Danu, you are Danuska, Danuska Balakrishnam.
Yes, doctor.
Yeah. Dila Nur Adila.
Yes, doctor. Yes, doctor.
Yes.
Doktor,
Mm-hmm.
exam sampai amino acid catabolism ke?
Yep.
Okay, thank you.
Alia Nadira, are you here? Saya tak jumpa kat sini.
Yes, doctor.
Oh, ada. Siti Aisyah? Siti Aisyah. Are you there? Okay. Nurin Liana? Okay, ada. Nur Aini? Nur Aini. Ada, okay. Terpinam mata saya tengok. Muhammad Aslam. Muhammad Aslam ada? Tak ada eh? Hanis Nazwa. Hanis. Tak ada juga.
Ada doktor.
Ada. Okay. Siapa lagi? Husna. Ada. Anu ada tadi. Nur Afrina ada. Siwaniswari. Okay.
Yes, doktor.
Okay. Sofita ada. Sofita ni saya perasan sebab dia selalu masuk awal. So she's always the first one to come into the class lah. Okay, well done Sofita. Okay, siapa? Nurul Ainun. Nurul Ainun. Tak ada. Shasmira. Tak ada. Nurul Afiqah.